All right, so how are you guys doing?
Good.
Good. Questions before we get started?
Not that I know of. How do they do the grading in that other thing that we do? The Parsons?
Pearson?
No, yeah, Pearson, sorry.
Um,
Like
oh, does it just show you points? I'll switch it so
it
it's
shows
percentages.
1 point for each, and then there was something where it was like 1 through like 80. So I was just trying to figure out like my grade for it, and then I tried to go back and see if I got anything wrong, but I couldn't figure it out. So
Oh, if you go to the grade book, how is it set up here? I'll go to it now without you guys seeing it.
yeah, I wasn't
We'll go to it.
sure because I wanted to see like if I got anything wrong. I wasn't sure how the grading goes.
Let's see. I'm still here. I'm just making it so you can't see everybody's grades because I don't want to do that.
Yeah, I just wanted to keep track and see if there was anything I needed to really focus on.
Sure. So in the grade book, you'll see there's grades for the introduction to mastering anatomy and physiology, homework, and the quiz. There's grades in there already for a lot of people. And then you can click on the actual grade in the grade book and it'll break it down by question. It'll tell you
Oh,
how many, what you got right, what percentage of each question you got right.
okay. I was afraid to start clicking things because I didn't want to mess something up. So I thought I would ask it. I'll just go on. I'll look at it later. I just didn't want to start clicking into things and then totally mess up the page and then because I want to make sure I don't
Right.
mess anything.
I do want to, that makes me question one thing though. I do want to check and make sure that I set it so that you can review everything. I thought I did, so let me just check that while I'm in here.
Because I know I went back in to go look and see if there was anything wrong and stuff like that, but it was just the quiz with my answers that I did with a check mark in the corner. So I didn't know.
Let's see. Allow rework for practice. Yeah, so you should be able to go back in and look at the questions and practice with them. It won't count for anything, but it should allow you to do that because it's set up. At least I've checked that box. So it should allow you to do that. So let me know if that's not the case. But it should allow you to go back in and rework the problems for practice. It won't show you what you got right and wrong. It was set so that it would only show you after the due date, but
Oh,
now
okay.
I changed it so that it'll show you after you hand it in. So you should be able to see, which ones you got right, and which ones you got
Okay. I'll look after.
wrong. Okay.
Thank you.
So what we'll do today is we'll talk about chemistry. And we'll finish anything up about chemistry that we need to on Friday, and then I'll make the test available to you guys on Friday. And I'll go into more details about how many questions it is and everything. It'll be somewhere around 40 or 50 questions, I think, something like that. But I'll open it up Friday. There'll be a time limit on it, so you can just go in once and do it. But you'll have all weekend. You'll have, if I open it up Friday, you'll have till like Tuesday to do it. Just whenever you can, whenever you have a block of time that's good for you. So I'll talk a little bit more about that after tomorrow because I want to use tomorrow to make sure I got everything set the way I want to and everything.
Okay. You said it's timed?
It's timed, right?
Okay.
So let me share my screen. And we will go to the PowerPoint for chapter 2. So were there any questions about chapter 1, about the anatomical terms, the you know, superior, inferior, the body segments, any of that stuff before we go on to chemistry.
Yeah.
So chemistry, the chemistry that we'll talk about today is very, very basic chemistry, right? We're not going to go into much detailed chemistry at all. But we do need to understand how these atoms bind together to really understand cells and tissues. So there are some basic things of chemistry that we have to talk about. So the body's most basic level of organization is chemistry. It consists of atoms and molecules. So even things that are not living are made up of atoms, right? Even plastics and metals and stuff are made up of atoms. But living cells are made up of atoms too. You know, molecules like glucose and other carbohydrates and lipids and proteins are made up of atoms. So chemistry is the science that investigates matter and its interactions. So you have to understand a little bit about chemistry, and then biology builds on that, right? Biology is the study of living things. Chemistry is just the study of all things, all matter. So if I asked one of you guys, what's matter? How would you define matter? What would you use to describe matter?
Is that the one that's anything that takes up space?
Yep, anything that has mass and takes up space. So it can be living or non-living, and that's what chemistry is investigating. And then biology kind of takes from that information and builds on it and just studies living things. Okay. So matter is anything that takes up space and has mass. Mass is weight, right? How much something weighs. And it's composed of substances called elements. So elements cannot be changed or broken down into simpler substances. An element would be something like oxygen or carbon or nitrogen or helium, right? It can't be broken down to anything smaller. If you have carbon, that's what you have. Whether you have 1 atom of carbon or 1 million atoms of carbon, carbon is carbon. And we'll get more into that in a little bit. So then we get into the atomic structure. So what's in an atom, right? There are little smaller pieces that make up an atom. And there's 3 of them in particular that we are concerned with in this chapter. And those are protons, neutrons, and electrons. Right? And over here on the right, you have a typical diagram of what one atom looks like. This happens to be an atom of helium. And it shows the protons, the neutrons, and the electrons. Okay? The protons and the neutrons hang out at the center of the atom. Center of the atom is called the nucleus. And when I think of an atom, I always compare it to a solar system. And so the nucleus is like the sun of the solar system, right? It's right at the center. The electrons are actually orbiting around the nucleus, right? So here are 2 electrons. They are traveling in a circular path around the nucleus. So I think of them like the planets orbiting the sun. And what you need to know, not only where are they located, but you need to know that protons have a positive charge, electrons have a negative charge, and neutrons are neutral. They don't have any charge. And that becomes important, and you'll see why in a few minutes. So in this diagram of helium, helium has 2 protons, right? So 2 plus charges. It has 2 neutrons, those don't have any charge. And it has 2 electrons, 2 negative charges. So protons and neutrons, they're the ones that hang out in the nucleus, they hang out at the center. They are similar in size and mass. They're about the same size. They weigh close to the same amount. Electrons are a lot smaller and a lot lighter. Electrons are the ones that are orbiting around in a circle, like the planets. And they are a lot lighter. They're much lighter and much smaller. So we'll get to the atomic number. So what I'm going to do right now is just go to your Moodle course. One of the things that I posted here in your chemical level of organization is the periodic table of elements. So I'm going to open that up for you. So here's a typical periodic table, right? This is actually a really simplified one. So before, when I said all mass can be broken down into atoms and atoms break down into elements, the elements are listed on what's called the periodic table. So these are all the elements that are known to man. They're always listed in the same order, and there are reasons, like there are reasons for these different colorations, and it's because there are similarities between the elements that have the same color. A lot of chemical similarities that we're not going to go into. Right? You can see that number 1 starts up here with hydrogen. You can go all the way to 118, right? It probably goes further than that now. This chart, this periodic table, is going to give you a lot of information when you are trying to determine different things about that element. For example, the atomic number is on the periodic table. So for each one of these elements, right, and each element is represented by a letter or 2 letters. For each element, there's a number above it, and that number is called the atomic number. And that number equals the number of protons of one atom of that element. So for example, C stands for carbon. Every atom of carbon has 6 protons. O stands for oxygen. Every atom of oxygen has 8 protons, right? And you can keep going all the way up to even the highest levels. Uranium, 92, right? Every atom of uranium has 92 protons. So this table literally is arranged by the number of protons, right? And that's how the elements are defined, by their number of protons. Carbon always has 6. If it's an atom with 5 protons, it's boron. If it's an atom with 7, it's nitrogen, right? If it's got 6 protons, it's always carbon. That's what defines the different elements. So back to your slides, right? The number of protons in an atom is the atomic number. The atomic number is always going to give you the number of protons. So for example, helium's atomic number is 2. All helium atoms contain 2 protons. There are these things called isotopes. Okay. Isotopes are atoms that have the same number of protons, but a different number of neutrons. Okay, and we're not even going to get much into what neutrons do because they're not really involved in the binding of atoms together. Okay, so just realize an isotope will have the same number of protons, but a different number of neutrons. The atomic weight, sometimes you'll hear of. On this particular periodic table, it doesn't have the atomic weight. On some of them below the letters, you'll have the atomic weight. Okay. The atomic weight incorporates the mass of an atom of that element. So it's adding up the mass of the protons and the neutrons and the electrons. Now remember, the mass of the electrons is really tiny, right? So the mass of the protons and the neutrons are each about 1, but the electrons are really, really tiny. That's where this 0.0079 comes in. Again, we're not going to get involved with the atomic weight. Make sure you know what the atomic number is. So, all right, electron shells. So I'm going to go back to this picture. This picture shows the 2 electrons of helium in an electron shell. Right? They are in a shell by themselves, orbiting around the nucleus. We will only study atoms that have 3 electron shells. right? You can get to atoms that are much bigger than that, have more shells than that. What you need to know is there is a maximum number of electrons that each electron shell can hold. So this electron shell, which is considered the first one, the one directly outside the nucleus, it can hold a maximum of 2 electrons. One outside that can hold up to 8 electrons before it's full, and the one outside that can hold up to 8 electrons before it's full. So 2 plus 8 plus 8, right, is the maximum number that we're going to go to. Now, we go back to the periodic table. Every single element that is on the periodic table has a net charge of 0. There's no positive, no negative charges. What that means is, let's take carbon as our example again. Carbon has 6 protons. What's the charge on a proton?
Positive.
Positive. So carbon has 6 positive charges. So how are we going to get it from 6 positive charges back to 0, right? We know the carbon right here, as it sits on the periodic table, is 0. Does not have a plus or minus charge on it. How are we going to get from having 6 protons? What do we have to do, or what do we have to have to make it get back to 0?
Have 6 electrons.
Good, 6 electrons. Why? What's
Because
the charge on each electron?
because the negative charge of the electrons counteracts the positive charge on the protons.
Perfect, exactly. So for every single element on the periodic table, this number up here literally equals the number of protons. But you also know now that that's the number of electrons of one of those atoms, right? It has to have the same number of protons and electrons to be 0, to balance out, so that you have the same number of positive charges and negative charges. So we get back to this electron shells, right? Again, atoms are neutral. Each positive proton is balanced by a negative electron. The electrons orbit the nucleus in shells. Only the electrons in that outermost shell, whatever the outermost one is, those are the only ones that can interact with other atoms. And the chemical properties of an element are determined by the number of electrons in that outer shell. So, think about all of this. There are 2 things that all atoms want in order to be as stable as possible. They want to fill their outermost electron shell. So like I said, the first shell, the one that's closest to the nucleus, holds only 2 electrons. The second one, the one outside of that, can hold up to 8. The third one can also hold up to 8. If an atom has an unfilled outer shell, it's going to try to react with other atoms to fill that shell, right? Atoms want their outermost shell to be full. They want to be neutral. They don't want positive or negative charges. And they want their outer shell to be full. Okay, so here is carbon, right, on the left. Carbon's atomic number is 6. So carbon has 6 protons and 6 electrons, right? Again, we're not going to worry about neutrons much for this class because neutrons don't do anything with the way these atoms bind together. But if we think about this, carbon has 2 electron shells. Oops. Carbon has 2 electron shells. How many can this first shell hold?
2,
2. How many can the second shell hold?
4,
How
8.
many can it hold maximum? 8.
right,
And
8.
how many does it have?
4 on there.
It only has 4, right?
Yep.
So what do you think carbon wants to be more stable? What do you think it needs?
Wants 4 more electrons in the outer shell.
It wants 4 more electrons in the outer shell. So carbon, one of the reasons why carbon is such a big part of everything in our world, in our lives, is because it reacts with so many different atoms, right? It needs 4 more electrons. So we can get 1 from 4 different atoms. It can get 2 from 2 different atoms. It can get 4 from 1 atom. It can react with a lot of different things. Over here on the right, what they are showing you is an atom of neon. Neon is considered an inert gas. Neon's atomic number is 10. So it has 10 protons and 10 electrons. So in the first electron shell, how many electrons are there? 2. In the second electron shell, how many electrons are there?
I'm sorry, that one, there's 8.
8.
8.
Yep.
So is neon happy? Yeah.
Yes.
Neon doesn't react with very many other atoms. It's really hard to get neon to react with another atom because it's stable, right? It's not something that's going to react with other atoms. So in order to fill that outermost shell and to be neutral, atoms will interact with each other. And those interactions form what are called chemical bonds. So these atoms interact with each other to form larger structures made of multiple atoms. And to do so, sometimes they share electrons so that multiple atoms feel like their electron shells are full, or sometimes they gain or lose electrons, right? Sometimes electrons will jump from one atom to another. What this causes are these bonds to form to hold the atoms together after the reaction is ended. This is a process that forms what are called molecules and compounds. Molecules and compounds are made of multiple atoms bound together. Okay, the big difference is that molecules contain more than one atom bonded together by shared electrons. So something like water or oxygen, right? O2, which you would find in an oxygen tank. Compounds are made up of atoms of 2 or more different elements in a fixed proportion. So water is a molecule and a compound. The biggest difference is that molecules are held together by sharing electrons, compounds not necessarily. In some cases, the atom is going to gain or lose electrons, right? It'll get to a point where, and we'll see this as an example, where an electron will leave an atom, right, and go to another atom. At that point, when an atom has lost or gained an electron, it's called an ion. At that point, it's going to be charged. And if it's a cation, it's got a positive charge. If it's an anion, it's got a negative charge. So let's think of an example. Sodium is a good example. Sodium's atomic number is 11. So how many electrons does sodium have?
11.
11. How many of those electrons go in its first shell?
2.
2. Good. How many go in its second shell?
8.
8. How many does that give you right there? That gives you 2 plus 8 is 10. We said we have 11. So that 11th electron has to go in a third shell all by itself. That doesn't make sodium real happy. So there are a lot of cases where that electron will leave sodium. Now, if that electron leaves sodium, and sodium is left with just 10 electrons, that means sodium still has 11 protons, right? Its atomic number is still 11. It still has 11 positive charges, but now it only has 10 negative charges. So now it's not balanced. It's not electrically neutral. It has a charge to it. And we'll get to that in a second. So bonds that are created between ions, those are called ionic bonds. And ionic bonds are created when that situation happens. When one atom gives an electron to another atom. One atom donates an electron to another atom. And the example that's most often given is with sodium. It turns out that chlorine, its atomic number is 17. So if you do the math out, it's got 17 electrons. 2 go in the first shell, right? So that leaves me with 15. 8 go in the second shell, that leaves me with 7. 7 go in the third shell. That third shell wants one more. So if you get an atom of sodium near an atom of chlorine, well, sodium really wants to get rid of one extra electron. Chlorine really wants to gain one electron. And that's what will happen. That electron will actually jump from sodium to chlorine. Now, the result of that is now you've changed the charges. Right now you have a sodium ion and a chloride ion. Because now you have a different number of electrons than you do protons. They're not balanced anymore. And what do we know about opposites? What do opposites do?
Attract.
They attract. So if you have an ion that's +1 and an ion that's -1, they're going to attract. And the +1 and the -1 cancel each other out. Right? So here's the example. Here it is in pictures. Here's your sodium atom. Right? Sodium has that 11th electron sitting out there in a shell all by itself. Down below that is your chlorine atom. Chlorine has only 7 electrons in its outermost shell. So it could really use an extra one. So this electron from sodium jumps to chlorine. And what you get is what's here in this middle panel, panel 2. Now, sodium up here, its atomic number is 11. So it still has 11 protons. But now it only has 10 electrons. Right? So it has 11 protons or 11 positive charges and only 10 electrons or 10 negative charges. So overall, this has a +1 charge. Right? So that's called a sodium ion. It's not a sodium atom anymore, it's an ion. If we come down to chlorine, its atomic number is 17. So it still has 17 protons, but now it has 18 electrons. Right? So it has one more negative charge than positive charge. So this is considered a chloride ion, chloride Cl-. It's got a -1 charge. And opposites attract, right? The plus and the minus, +1 and -1 attract each other, and the sodium and the chlorine bind to each other pretty tightly, and that gives you sodium chloride. Now, sodium chloride is table salt, right? It's a compound that exists, and it's called an ionic compound, right? This is called an ionic bond, and it's because there's 2 ions binding together. So the movement of the electron is not what caused the bond. Movement of the electron caused the charges. The charges cause the bond. Questions on that? So one way that atoms bind together is through an ionic bond. And an ionic bond is always where one atom donates electrons to another atom, right? One atom is losing electrons, another one is gaining. But sometimes you have a bond that forms where atoms share electrons. So in this case, or in these cases, one atom is not losing any electrons, one atom's not gaining any electrons, but they're both sharing the electrons, right? They both feel like they have all these electrons. Those are called covalent bonds, right? So ionic bonds is where the electrons move. A covalent bond is formed when there is the sharing of electrons. These are really very strong bonds. These are the strongest type of bonds. They're much stronger than ionic bonds. And there are single covalent and double covalent bonds where a single covalent bond is sharing of one pair of electrons, a double is sharing of two pairs of electrons. But just to give you an idea, right? Oftentimes when you see hydrogen or oxygen in a tank, for instance, it'll say H2, right? Or it'll say O2. It's because one atom of oxygen is not really all that stable. Oxygen's atomic number is 8. Okay, so it has 8 electrons. So you put 2 in the first shell, and you put 6 in the next shell. That shell wants 8, right? The shell wants 2 more. So oxygen by itself is not real stable. If you get 2 atoms of oxygen together, though, and they're real close to each other, they are sharing these electrons in the middle. So both of these oxygens feel like they have 8 electrons in their outermost shell, even though neither of them do, right? And O2 is very stable. If you know anybody with oxygen tanks, right, it's always O2. That's the stable compound or the stable molecule. And so here they're showing the same thing with hydrogen, right? Hydrogen is number 1 on the periodic table. It has 1 proton and 1 electron. Well, having 1 electron doesn't make you all that stable. You want 2 electrons in that shell. You get 2 atoms of hydrogen together. They can each share their 1 electron, and each of them feels like it has 2 electrons. Remember, these electrons are moving. So we're drawing them in stationary places, but they are like planets orbiting around the sun, right? They're constantly moving. Now, when we talk about covalent bonds, there are some that are polar and some that are nonpolar. When I say the word polar to you, and having nothing to do with science necessarily, what do you think of when you hear polar?
Cold.
Cold, good. What else? Bears? Nobody says bears? Most people say polar bears. So cold, you think of the North Pole, right?
Cold, Arctic, yeah.
Arctic, good. Well, it turns out the Earth has a North Pole and a South Pole, right? We have magnetic north and magnetic south. Polar really means that there are 2 opposite ends. 2 ends are different, right? In the case of the Earth, there's north and south. Batteries, if you have a remote control for your TV and the batteries die, does it matter which way you put the new batteries in? Yeah, right? You have to look at the inside of the little case and see which end is positive and which one's negative. Batteries have a positive end and a negative end. Batteries are polar. They have opposite ends. Anything that has opposite ends is polar. Sometimes you hear about a celebrity or a politician that's polarized. What does that mean if they're polarized? They have a polarizing personality.
Like a split personality.
No, not a split personality.
'Cause I
Like
feel
people either, like
There's
people
more-
either love them or hate them.
Exactly. It has more to do with how people perceive them. People either love them or hate them, right? The example I'll throw out there is Donald Trump. Right? People feel very passionately one way or the other about him. Nobody's in the middle about him, right? He polarized the country. He made part of the country loves him, part of the country hates him. So polarizing means opposites, right? It means that there are two different ends. The same thing is true when you're talking about molecules bound with covalent bonds. Some of these molecules have two different ends. They have ends that look very different. Those are polar covalent. Some of these molecules have ends that look identical, right? You could flip them around, they'd be identical. Those are non-polar covalent bonds, okay? So non-polar means the ends are the same. Polar means the ends are different. Now, I'm going to show you, give me a good one. Okay. So I'll go back to this. Is H2, is that polar covalent or non-polar covalent?
It looks non-polar.
Non-polar covalent, right? The ends are the same. Each end is an H. They're each pulling with the same amount of force on those electrons in the middle. Same thing with O2, right? It's non-polar. Both of these oxygens are the same size. They're both pulling with the same force, equal sharing of electrons. Even something like carbon dioxide, which is a carbon in the middle with an oxygen on each end. Both ends are the same. They're pulling and they're sharing these electrons in the middle with the same force. Now we get to something like water. Okay, and ignore the hydrogen bond stuff on the left. We'll get to that in a minute. What's the chemical formula for water?
H2O.
H2O, right? Everybody knows that. So what that means really is that one molecule of water is made up of two hydrogens and one oxygen. Now, the atomic number for oxygen is 8. The atomic number for hydrogen is 1. When I first started telling you how to draw an atom, how to draw a representation of an atom, I compared an atom to the solar system, right? And I said the nucleus is like the sun, center of the solar system. The nucleus and the sun, right? The sun has some sort of gravity, and that's what keeps the planets orbiting around it. That's what keeps us stuck to the planet. The bigger the sun is, the more gravity it has, right? The more pull it has. Now, if I'm looking at water, if I'm looking at this molecule that is made up of 2 hydrogens and 1 oxygen, it's 3 atoms, right? So there are literally 3 suns. There's one at the center of oxygen. There's one at the center of each hydrogen. Okay. Are all 3 of them the same size? No. Right? Irene's shaking her head no. Good. Which one is the biggest?
The oxygen.
Oxygen. Oxygen has 8 protons, right? So the center of oxygen is much bigger than the center of each hydrogen. Each hydrogen only has 1 proton, right? So the center of oxygen is much bigger. What that means is the sun at the center of oxygen is bigger and is going to have more gravity. It's going to pull things towards it more often. Okay, everybody following me? So remember, the electrons are orbiting around the sun. And in the situation of water, it's a covalent bond. So the hydrogen is, each hydrogen is sharing electrons with the oxygen. Since the oxygen has a bigger nucleus, a bigger sun, the electrons are going to spend more time circling around the oxygen than they will around either hydrogen, right? They may orbit oxygen 10 times and hydrogen once, and 10 times and then once. Does that make sense? Right? It's unequal sharing of electrons. The electrons are not being shared equally between one hydrogen, the other hydrogen, and the oxygen, they are spending more time around the oxygen than they are around either hydrogen. It's polar, okay? The ends are different. The ends look different. They're not sharing the electrons equally. The result of that, what's the charge of an electron?
Negative.
Negative. These negative charges are hanging around oxygen more often than they're hanging around either hydrogen. Result of that is that this end of the molecule has a little negative charge, right? And you can see that written there. Whereas the hydrogens have a little bit of a positive charge. And it's because the electrons are spending more time around oxygen. The negative charges are spending more time here than they are over here. So the molecule itself acts like this end is slightly negative and this end are slightly positive. That's because it's a polar covalent bond. That's because there's not equal sharing of electrons. It's because one atom is pulling the electrons with more force than the other atoms. Right, so the electrons, which are negatively charged, are hanging out at one side of this molecule more than the other side. And I'll flip back to this slide. So polar covalent bonds happen when there's an unequal sharing of electrons. One end has a slightly negative charge, one end has a slightly positive charge. And it's because the molecules, the electrons, excuse me, are hanging out at one end of that molecule more often than the other end. Good example of that is a water molecule. Now, what that causes is something really cool, and it's the third type of bond, and these are called hydrogen bonds. So we had ionic bonds, we had covalent bonds. Now we've got hydrogen bonds. Hydrogen bonds occur between molecules. So ionic and covalent bonds occurred between atoms to make molecules. But whenever you get a molecule like water that's made by a polar covalent bond, you get kind of a negative charge at one end and a positive charge at the other end. And what do we say opposites do?
Attract.
They attract. So the positive end of this water molecule is going to attract to the negative end of another water molecule. The negative end of this water molecule will attract to a positive end of another water molecule. So these dotted lines, these red dotted lines, represent bonds called hydrogen bonds. Okay, it's a slight attraction. It's the weakest attraction. Of the three types of bonds we talked about, this is the weakest. So covalent's the strongest, then ionic, then hydrogen. It allows for this hydrogen bond to happen between water molecules. Water molecules stick together, right? There's surface tension on a lake. We've all seen like a bug land on a lake and it just sits there, it doesn't sink, right? Well, its weight is distributed so far out that it doesn't even break the hydrogen bonds. It's not heavy enough to break the hydrogen bonds. It's not heavy enough to break these dotted lines. Okay, and part of what we'll go through in the next part of this chapter is our unique characteristics of water, why water is so important to life. But a lot of it has to do with these hydrogen bonds. Now, the example that we're using is water for hydrogen bond. There are other molecules that hydrogen bond. Okay, this becomes a really large, important bond in holding DNA together. So DNA is double-stranded, right? And those two strands are held together by hydrogen bonds. It also is a really important bond in terms of making three-dimensional proteins. So all proteins, their most important thing is their 3D shape, and that is in large part due to hydrogen bonds. So hydrogen bonds don't only occur between water molecules, even though that's what we're going to talk about, and that's the example we're using. They occur between any molecules that are made by polar covalent bonds.
So whenever there's a molecule with a larger, like an atom that's much larger than the other atoms, the electrons are more drawn to the larger atom and it's always polar. Like, is that kind of a rule?
Yes, only if it's covalent bonds, right?
Okay,
Remember, ionic bonds, the electrons actually move. So
right.
you don't have this.
Gotcha.
But it's whenever they're sharing electrons, the electrons are spending more time around the larger atom than the smaller atoms. And so it creates a situation where there's kind of this difference between the two ends. One end is more negative, one end is more positive. Right. And so that leads to surface tension, right? Attraction between water molecules forms surface tension. Again, we've all had, all seen bugs land on water. If you take a glass out of your cupboard, right, and you start filling it up with water and you aren't really paying attention, you can go past the edge of the glass with water. You can go past the top and it won't spill right away. It won't spill until you get way over the top. You can kind of get a hump, a curve over the top of the glass of water because the water molecules are sticking together and gravity hasn't pulled them apart yet.
Is there kind of a similar thing going on with like viscosity and those kinds of like liquid situations?
With viscosity. So depending on what the molecule is, yes. And with water, what we'll see is that there's something called adhesion and cohesion, where the water molecules stick to each other and they stick to things around them, like the inside of your blood vessels. And so that's what allows things to move through our blood vessels or through a straw. So yes. Now, what I should have done is taken out these slides. What I can do is take these slides out and I will email everybody. We're not going to talk about reactions right now, synthesis and decomposition reactions and enzymes. We'll talk about those at a later date. Okay. But I want to go right on to properties of water. So there's a few slides in there on synthesis reactions and decomposition reactions. I'm going to delete those and re-upload this slide deck. And I'll email everybody, since there's only a few of you on the call here, to make sure that everybody knows that I deleted those slides. But what I want to talk about next are the properties of water. So we all know water is the most important substance in the body. You hear it all the time, right? You hear you need to drink water. It makes up most of your body. Well, why? Well, there are some general properties that make water really important to the body. One is that it's an essential reactant in the chemical reactions of living systems. One is that it has a high heat capacity. One is that it's an excellent solvent. And we'll get to those, right? So chemical reactions in our body take place in water all the time. So when you eat any foods, right, you're usually eating large molecules and your body wants to break those molecules down to get the individual nutrients from it. And in order to do that, you have to add water, right? It's a process called hydrolysis, where you're actually adding water to break that molecule apart. We'll get into that in another chapter. But water is really important because it's necessary for all those reactions to occur. Okay. When you are linking two small molecules together, you're actually pulling a drop of water out. When you are breaking apart two molecules, you're putting a drop of water into it. So water becomes something that is necessary for most of the reactions that occur in our body, most of the chemical reactions that occur in our body. And I'm thinking especially of your digestive system, but even your cells are making proteins, right? Because proteins are what we need to function. And making those proteins happens because of water, right? You can't actually link together those amino acids without water. Water has a high heat capacity. It can absorb a ton of heat, right? Think about it. Summer just ended, or is just ending now. But you could go outside in the summer, you could go to the beach and spend the day at the beach or at the pool, and your body doesn't burn up, right? As a matter of fact, your body temperature barely goes up at all. And it's because there is so much water in your body that the water is absorbing that heat. It acts as a heat sink, right? It absorbs the heat so that your liver doesn't cook and your intestines don't cook. And none of that stuff heats up because the water is just absorbing the heat, right? And as the water absorbs the heat, it evaporates. And that's why we need to constantly drink more water when we're out in the heat, because we're losing water. But it has this insane ability to hold on to heat. So the opposite of it is true as well. How do we cool off? What do you do to cool off? Generally, what does your body do?
Sweat.
Sweat. Anybody know how sweating cools us off? So all sweating does, right? You get little droplets of water, basically, on our skin. That doesn't cool you off. What cools you off is those droplets turning to air. They evaporate. And when they evaporate, they take a lot of heat with them. It takes a lot of heat to get that drop of water to go from a liquid drop to air, right? To a gas. So that's why sitting in front of a fan works, right? The fan will evaporate those water droplets and cool you down. If you say you go to the gym, you get in the car to go home, you crank the air conditioning. It gets cold really fast, right? Your shirt will actually get cold to touch. And it's because it not only does water absorb a lot of heat, but it takes a lot of heat to go from a drop of water to a gas, to a steam, a vapor, whatever you want to call it. And so it's taking a lot of heat with it. So not only does it act as a good heat sink when we're out in the sun, but it can cool us down really quickly. And all this has to do with these hydrogen bonds, right? It takes a lot of heat to break those hydrogen bonds, but once you do, all of a sudden that heat dissipates. So water is necessary for chemical reactions in our body. Water has a high heat capacity. Water is great at being a solvent. So if you take a spoonful of sugar and you put it in a glass of water and you stirred it up, what happens to the sugar? It's still there?
It dissolves.
Dissolved, right? Same thing if you take a spoonful of salt. Take some table salt, put it in a spoon, stir it into a glass of water, it will dissolve. Well, the reason it does that is because water has those slight charges. Remember, water has one end that's kind of positive, one end that's negative. If you think back to what salt is NaCl, right? We said that these two atoms are held together because one is positive and the other is negative. Well, water has the ability to pop off the positive sodium and pop off the negative chlorine and start one by one pulling those atoms apart. And that's how it dissolves things. So it becomes a really good solvent. I wish I had a picture of that. It becomes a really good solvent. Pretty much anything that is bound by an ionic bond can be dissolved with water, right? Because water can take the positive and the negative charges and pull them apart. In intro to bio class, we talk about, Water is what allows things to move through our vessels. We talked about the effect of something like a straw, drinking straw, how that works. Think about trees that are outside, right? There are plenty of trees that are 50 feet tall, and they have leaves all the way at the top of them that are green. What do plants need? What do trees need to keep their leaves green?
Sunlight and water.
Sunlight and water. Good. Where do plants get water from?
The soil from
Soil,
rain.
the ground, right? The roots.
Your-
So if you're talking about a leaf that's 50 feet up in the air, it's getting its water from the ground. So you've got from the roots going all the way up to that leaf that's at the top of the tree, you've got a really long tube, like a really long straw. So what happens during the day is those leaves have little tiny openings, and they open up in the sunlight, and the sun causes a drop of water to evaporate. When it does, it pulls a whole chain of water molecules behind it. There's a whole chain of water molecules going all the way down the trunk to the roots. And so when you pull on one at the top, that whole chain moves. And that's how it pulls water up from the ground. That's the same way that a straw works, a drinking straw. Okay, so water has all these really cool properties because of these hydrogen bonds. Now we get to pH, right? We talk about pH a lot of times with the body. What is the pH scale? What numbers does it run from? Anybody know? pH scale runs from 0 to 14, right? 7 is neutral. Anything between 0 and 7 is acidic. Between 7 and 14 is basic. So when you talk about pH, when you write pH, you always write it with a small p and a capital H, and there's a reason for that. What was the chemical formula for water?
the
Oh.
H2O.
H2O, right?
Mhm.
Can I just as easily, that means that there's 2 hydrogens and 1 oxygen. Can I just as easily write that HOH? Right, that's still 2 hydrogens and 1 oxygen. In any solution, a really tiny, tiny, tiny amount of water dissociates. And when it dissociates, it breaks apart into H's and OH's. Right? H's are called hydrogen ions, OH's are hydroxide ions. Now the 14 is a really weird thing because it has to do with negative exponents. I'm not going to get into all that. But just realize if you have a solution with water, with HOH, and it breaks apart into even amounts of H and OH, that would be 7H and 7OH. Okay? What you are measuring with pH, that capital H, is the amount of H. When water breaks apart into H and OH, pH scale is really measuring how much H there is. If it breaks apart into even amounts, you end up with 7 H's. It has to add up to 14. Again, I'm not going to go into the reasons why. If it breaks apart so that you have more H's, because it's a negative exponent thing, your H would be something like 2 or 3 or 4, and your OH would be 12 or 11 or 10, would always add up to 14. The whole point of this is that what pH is really measuring is when that small, tiny, tiny amount of water breaks apart. How much of it, what percentage of it breaks into H's, right? Is it a 50-50 percentage, or is it more H's, or is it less H's? If there are more H's, it's acidic. If there are less H's, it's basic. Okay. So pH represents the concentration of H hydrogen ions in the solution. The higher the concentration of hydrogen ions, the lower the pH. This is because it's taken from a negative exponent. So it's h raised to the 0.000000 something, which we don't have to get into. But any solution with a pH of 7 is neutral. As you get closer to 0, it's more acidic, right? So 5 is slightly acidic. Something like a 2 or a 1 is very acidic. If you go the other way on the scale, it becomes basic or alkaline. These two words, basic and alkaline, mean the same thing. So again, something that is 9 is slightly basic or slightly alkaline. Something that's 13 is a very strong base or a strong alkaline solution. Okay, and so here is kind of just a scale, pH scale with some examples. Pure water would be neutral, right? Like distilled water, no ions or anything in it, would be neutral, be about a 7. Notice that things which are acidic tend to be foods and drink, right? Things that we eat and drink tend to be acidic. Look at how low hydrochloric acid is. It's probably closer to 2, but extremely acidic. Stomach acid is close to battery acid in the grand scheme of things. That's kind of an anomaly. That's something that, you know, not usually in the body. Most things are here, right? But stomach acid is way down here. As we go on the neutral side, right, you get to more cleansers. The more basic you get to, the more cleansers you get to. So things like soaps would definitely be somewhere in the slightly basic range. Bleach and ammonia are much more basic. Oven cleaner is kind of the most basic thing, or Drano, right? If you pour that down your drain to clean out the any of the, you know, hair or anything that's in your drain, that would be the most basic. So again, you should just know what the pH scale is, know some examples of it, examples that are on each end of it. Most bodily fluids are just over neutral, so 7.35 to 7.45. Most things like blood is here, saliva is a little bit more acidic. Hydrochloric acid, stomach acid is the exception. That's really acidic.
Actually, hydrochloric acid in your stomach only has a 0.012 molar solution, which is really weak.
Right, but the pH of it is extremely acidic, which is why if you throw up a lot, it actually, you know, it burns your your esophagus and your mouth.
Huh. They didn't mention that. I watched his YouTube channel, Institute of Human Anatomy, and they were talking about the hydrochloric acid in your stomach.
Oh,
And how, yeah, for some reason my camera's not working.
oh,
Let me see what's going on here. I think it's my streaming platform because it's on.
so you're right. It's not very concentrated, but it's it is acidic. It's
Is that
um,
possible?
so it probably wouldn't burn like the outside of your skin,
Yeah.
that kind of thing, but it would, you know, but it definitely does damage to your esophagus and your mouth, the inside of your mouth. So there you are. All right. So then we get to, right, organic compounds. So the rest of this chapter is on organic compounds. So if a compound is organic, what does it contain? Who knows? In terms of biology, chemistry. Anything that's organic contains carbon, right? So we use the word organic sometimes at the grocery store or something, and it means something different there. It means produce was grown without any, you know, pesticides or any chemicals being sprayed on it. But organic in chemistry or organic in biology means it contains carbon. And so there are 4 big groups of organic molecules that we have to get, or that we need for life. And they are carbohydrates, lipids, proteins, and nucleic acids. Now, Can we make our own carbohydrates? What do you think?
No,
No, we can't, right? Plants can. What can plants do that we can't do? What's that process called?
is it photosynthesis?
Photosynthesis. So plants can sit out there in the sunlight and take energy from the sunlight and link together a bunch of carbon dioxide molecules and make these carbohydrates. They can make sugars, which is really cool. We can't make sugars. We have to eat plants, or we have to eat animals which ate plants. Right? That's where we're getting our carbohydrates from. So carbohydrates are a good source of energy. That's what we use for energy. These include sugars and starches. There are 3 major types, and this is just based on the length of the actual molecule. Monosaccharides are just 1 subunit long, right? They're just the simple sugars. If you link 2 monosaccharides together, you get a disaccharide, which is 2 sugars long. And if you keep linking them together, you get a polysaccharide, which is a chain of sugar. So here are some different ways to draw glucose, right? You can draw it with the carbons going down the middle and each atom that's linked to it. More commonly, you see glucose drawn in a ring form like this. Oftentimes, they don't even put the C's here. It's understood that the corners of each of these ring structures have a carbon. But glucose is what we need for fuel to make energy. So that's why that molecule is important. If you link 2 monosaccharides together, it's called a disaccharide. For example, sucrose, table sugar, is a disaccharide. They are very soluble in water, right? And they have to be broken down into their monosaccharides before we can get energy from them. And so here you can see linking glucose and fructose together, right, to form the disaccharide sucrose. Right? Or just the opposite, breaking apart sucrose, which is what you would do in your body, into glucose and fructose, and you'd be using the glucose for energy. What you have down here are a bunch of glucose molecules linked together to form glycogen. Now, glycogen is a polysaccharide. It's a chain, right? You can see from here that it's literally thousands of glucose molecules linked together. So there's 3 chains you should really know. There's 3 polysaccharides you should know: starch, cellulose, and glycogen. Starch is how plants store glucose, right? So plants are sitting out there undergoing photosynthesis. They're making glucose, and they're storing that as starch. They're linking those molecules together and storing them. Then when you eat that plant, when you eat that fruit, you're getting the starch from it. And you're hopefully breaking it down into its individual glucose so you can use it for energy. Glycogen, which they call animal starch. So when you eat a meal, you almost always eat more starch than you can possibly use at that moment for energy. So you're going to have some glucose left over. So what your body does is it links some of those glucose molecules together as glycogen, and it stores it for later, okay? It's usually stored in the liver, stored in some of the muscles as well. But basically, you eat a meal, it's got more sugar than you can use at that moment. So your body knows, okay, let's link these extra sugars together and store them for later. When I don't have any sugar readily available, I can pull that out and use that for energy, right? Insulin is in charge of doing it. Then we have this third example of a polysaccharide called cellulose. This is a component of plant cell walls. They use it strictly for structure. It cannot be digested by humans. We have no enzyme to break it down. It's not bad, it's not good, right? It's just neutral. The best way I can kind of illustrate this, think of a piece of celery, Right? We've probably all eaten like celery with peanut butter in it. Bite a piece of that celery. Does it just break off as a nice crisp bite? Or you almost always have those little strings, right? Those little strings of celery. Those are cellulose molecules, right? Those strings are actually what the celery plant makes in order for that structure to be really strong, that stem to be really strong. We can't break that down. We have no way of breaking that down. So it's not good or bad. It's just kind of neutral, right? You can eat as much celery as you want. All you're getting from it really is water. You're not getting any nutrients from it. You're not getting any calories from it. Okay. So carbohydrates, again, these are used for energy. Lipids are used for energy storage. Right? They're important as energy reserves. Oftentimes, if you eat more carbohydrates than you can use at that point, some of those carbohydrates are converted into fatty acids and stored that way in your body. There are places in our body where we have lipids that serve as kind of an insulator or a cushion, right, around certain organs. Lipids are insoluble in water, right? They're the only one of these 4 groups that are not soluble in water. So you could take something like oil, you could mix up oil and water in a bottle all day. As soon as you set it down, those two are going to separate, right? Lipids are insoluble in water. It's why we use things like wax on our cars, right? You put wax on the car so that when water hits the car, it beads up. It doesn't do any damage to the car's surface. Okay, down here, there are the major types of lipids. Fatty acids and fats are what we're talking about for energy reserves. Steroids, we'll talk about, there's a whole chapter on steroids. Those are a type of hormone, right, made by the endocrine system. Phospholipids are a very specific type of lipid that make up the membrane of every cell. So we have trillions of cells in our body. So I don't want you to concentrate on steroids or phospholipids, but concentrate on fatty acids and fats. Those are used for energy reserves. And here you can see a fatty acid, right? This happens to be lauric acid. Fatty acids can be saturated or unsaturated, right? Saturated tends to be worse for, health reasons. It's worse. Saturated literally means that every single carbon is bound to as many hydrogens as possible. So every carbon is saturated with hydrogens. It's bound to as many as possible. If it's unsaturated, you've got some double bonds in there between carbons. So not every carbon is bound to as many hydrogens as possible. It's unsaturated. So like monounsaturated means it has one of these double bonds. If it's polyunsaturated, it has multiple double bonds, right? So anything that's unsaturated tends to have more of a bend in it, more of a wave in it, and tends to be a liquid at room temperature. Better for you health-wise. Most fatty acids end up attaching to a glycerol, forming what are called triglycerides. So 3 fatty acids, and this is the glycerol group. And these are the molecules that serve as energy reserves, but also insulation and cushioning in the body. Again, don't worry about steroids. They are a lipid, but we'll talk more about steroids in a different chapter on the endocrine system. And phospholipids, don't worry about phospholipids. We'll talk about those with cell biology. Those both are types of lipids, but don't worry about them right now. Okay, so then we get to proteins. Proteins are the most abundant organic molecules in the body. They do everything in the body. They will carry out pretty much all the functions in the body. Proteins proteins are enzymes. So all enzymes in the body are proteins, even though we have proteins that do other things as well. So here are a bunch of different categories of protein functions in the body. So just like with the different types of lipids, right, there are different types of proteins. What I really want you to concentrate on are the ones that are involved in metabolic regulation, okay? those are the enzymes. So we'll see that, you know, we make proteins for support. There are proteins that support like our muscles, right? Movement, same thing, actin and myosin help us move our body. Transport, there are proteins that we'll learn about that allow us to get things in and out of cells. Buffering involves proteins that will help us keep the pH constant. Defense, antibodies are proteins. So we have white blood cells that make certain white blood cells that make proteins to defend our body against disease. But what I really want you to concentrate on are metabolic regulation. And what's important for all of these types of proteins is their shape, their three-dimensional shape. So a protein is made by linking together amino acids. So you get this long chain, you get this, you know, think of it like beads on a string. And then that chain folds up. And it folds up in a very specific manner. And that is the most important part of all proteins, is their three-dimensional shape. Okay, so here goes through just what some of the different proteins are. Again, I want you to concentrate on number 5, the metabolic regulation, working as enzymes. Concentrate on that right now.
You
All
okay?
proteins, hello? All proteins are made of amino acids linked together. Amino acids, there are 20 different amino acids in the body. They all look very similar. And here's a diagram of one of the atoms that are found in one amino acid. They all have this group in blue, which is called the amino group. They all have a carboxyl group, which is in pink. They all have the central carbon, right? And a hydrogen coming off of that central carbon. Where they differ is this R group. This R group is different in all 20 amino acids. So all 20 of them look the same on these 3 places, right? But they look different where that R group is. And so here are just some different amino acids, right? Glycine, alanine. And what they do is they link them together. So you just keep linking more and more amino acids together. But the most important thing about proteins is their structure, their three-dimensional structure. And so there are 4 levels when you're talking about structure. There's primary, secondary, tertiary, and quaternary. Okay, so first, second, third, and fourth structure. Primary structure is just the sequence of amino acids linked by peptide bonds. Just a straight line, right? You're just linking those amino acids together. Secondary structure is where you start to get some shaping of that line, right? It usually creates either a helix or a beta sheet. And then tertiary structure is where you get much more complex coiling and folding into a true 3-dimensional shape. So the tertiary structure is what's incredibly important. Quaternary structure is interaction between multiple tertiary structures. So some proteins are actually made of like 3 different subunits. Each subunit is a tertiary structure, and then they're linked together. That linkage together is called quaternary structure. Okay, and so here it is in pictures, right? Primary structure up here, just a straight line, just amino acids linked together. They start to form secondary structure. So what you start to get is some bending. Now, when you get this bending, all of the sudden, some of these R groups are closer to each other than they would have been in a straight line. And so they start to react with each other, and you get much more complex folding and bending, which leads to tertiary structure. Okay, and then they give the example of hemoglobin here. Hemoglobin is made up of 4 subunits. So you can kind of see 2 of the subunits are this purple color and 2 are this blue color. There are these 4 subunits. Each of those is tertiary structure. Together, they're called quaternary structure. Again, all these different classes of proteins that are based on their properties, don't worry about them right now. but understand that the protein function depends on its shape, right? And that shape can be changed. There can be a change in the three-dimensional shape of a protein. It's usually caused by things like really high or really low temperatures, or really high or low pH or salt concentrations. And what happens is the protein basically unfolds. And when it unfolds, it becomes non-functional. And that can cause damage to organs or organ systems that you may or may not be able to even repair. The reason that this three-dimensional structure is so important is that in an enzyme, you've always got a part of that three-dimensional structure that's called the active site. And that's where something will bind to the enzyme. And that something is always called the substrate. That's what you start with. And binding it to the active site of the enzyme changes it into a product. So it's always taking something and creating something else with it, right? And what's really important for that is that three-dimensional shape. So here we have an enzyme in blue down here. The active site is highlighted in purple, right? So if this protein is not folded correctly, its three-dimensional shape is off just a little bit. All of a sudden, this active site is shaped differently, and it won't bind to S1 and S2, whatever those are, right? But as long as the three-dimensional shape of this enzyme, this protein is fine, it will bind to S1 and S2. Now you can see when S1 and S2 are near each other, they bind together. So you go from substrate 1 and substrate 2 to one product, and that gets released. Nothing has happened to the enzyme. As a matter of fact, this looks the same as it did over here in this first picture, right? So now the enzyme can bind to another S1 and another S2 and make another molecule product. And that's how enzymes work. Enzymes just speed up the reaction, right? Without an enzyme, S1 and S2 might never find each other. They're floating around in the body. They might never find each other, but they're both going to bind to the enzyme. Then they're right next to each other and they can react. And that's what enzymes do. Okay. The last thing to talk about are nucleic acids. And I know this chapter is kind of long, so that's why I'm kind of hurrying a little bit. Nucleic acids are, again, long chains, macromolecules, big molecules. They store genetic information. So things like DNA and RNA. Okay. DNA passes on genetic material from your parents to you or from you to your children. RNA is how we get information from that DNA to make proteins. Okay. The structure of nucleotides. So we saw that proteins were made of amino acids. There are 20 amino acids. Nucleic acids are made of nucleotides. And there are actually only 4 of them. There's only 4 nucleotides. All of them have a sugar and a phosphate group. That's the same in all 4. What's different is this nitrogenous base, this blue group. And there are, in DNA, there's adenine, guanine, cytosine, and thymine. Those are the 4 nitrogenous bases. So you'd have A's, G's, C's, and T's. One of the big differences between DNA and RNA is that in RNA, you don't have T's, you have U's with uracil. But you still only have 4 letters that make up RNA. So DNA is made up of A's, G's, C's, and T's. RNA is made up of A's, G's, C's, and U's. Other differences, RNA is a single chain, right? So here's an RNA molecule. This tan line here represents the sugars and the phosphates, and then the nitrogenous bases are what are sticking off of that. A DNA molecule is essentially 2 of these RNA molecules slapped together. So it's almost, to me, it looks like a ladder, right? Like 2 sides of a ladder, and the rungs are the bases. And then in DNA, somebody has twisted that ladder, right? So now it's this DNA double helix. So you can see here's one side of the ladder, here's the other side of the ladder, and the rungs of the ladder are the bases that are being held together. And by the way, these bases are being held together by hydrogen bonds, right? That's what holds A's to T's, is the hydrogen bonds, or C's to G's. The other molecule that I want to talk about with nucleic acids is something called ATP, because we'll talk about this a lot this semester. So instead of an A, a G, a C, or a T, or a U, sometimes we are making a nucleotide called ATP, and that stands for adenosine triphosphate. And so here is the molecule down here. Adenosine, or adenine, I should say, is the nitrogenous base. When you link that with ribose, it's called adenosine. And then, so that's the A. The T and the P stands for triphosphate. You've got 3 phosphates right here, right? 1, 2, 3. This is the molecule that we use for energy. We have cells, we have structures within cells that can take glucose and oxygen and make this. Okay, they actually pull apart the glucose, and they add on oxygens in a certain way, and through biochemistry, they make ATP. This is the molecule that all cells use for energy. So this is the molecule that your muscles are using to contract. It's also the molecule that's used to move things in and out of a cell. Right? What you need to realize is that with any molecule, what's holding these things together are bonds that contain energy. So you've got these 3 phosphate groups. And how do you keep them stuck together? Well, you stick them together with glue, with energy. And then when you need that energy, you break that bond and you release the energy. And that's exactly what we do with ATP. When we have a molecule of ATP, we actually break this bond between the 2nd and the 3rd phosphate. And you end up with a molecule called ADP, adenosine diphosphate, and a 3rd phosphate. But what you've done is you release the energy that you can then use to do some sort of work. I want to make sure to talk about this molecule a little bit, because this is what we're going to refer to kind of all throughout the whole semester, right? The reason that we have our digestive system, at least part of the reason of it, is to gain glucose so that we can make ATP for energy, right? When we talk about our respiratory system, it's so that we can get oxygen into our body, so that we can convert oxygen and glucose into ATP. When we talk about our circulatory system, it's how do we get that oxygen from our lungs to our cells to make ATP, right? So we're constantly trying to make as much of this energy as possible. And I will refer to it a lot this semester. So I want to make sure that you understand that this is a nucleic acid. This is just one, it's not a big long chain, it's just one link, right, of the chain of a nucleic acid. All right. So I know that today was a lengthy lecture. There's a lot in there. But we went through most important things, right? We learned about an atom. We learned about protons, neutrons, electrons, how to draw an atom. We learned that atoms bind together in order to have a neutral charge and have their outermost shell full. We learned about ionic bonds and covalent bonds. Ionic is where electrons are donated. Covalent, they're shared. We learned about hydrogen bonds, which stick together things like water molecules. It makes water unique, has a lot of really cool characteristics. We learned about pH a little bit. You should know what the pH scale is, what some acids are, what some bases are. Right? And then we learned about carbohydrates, lipids, proteins, nucleic acids. Carbohydrates are used for energy, right? Hopefully you can see from that ATP molecule that we're always trying to break starch down into glucose so that we can make ATP from it. Lipids, I said, focus on energy storage with lipids, right? Energy storage and cushioning and insulation. With proteins, I said, focus on enzymes. We talked about enzymes and three-dimensional structure. And then with nucleic acids, we talked about a little bit about the differences between RNA and DNA. We finished up talking about ATP. So, plan going forward. We'll meet again on Friday. We'll discuss any questions. We'll go over things again. I'll see if I can come up with a little bit of a lab thing, but I'm almost thinking just giving you some more. I'll come up with something to kind of give you some more to study for chemistry, to be able to study the chemistry, because this is such a long chapter. I will make the exam available on Friday. Okay, and so I'll know the details of the exam on Friday. I just want to finish it, look over it tomorrow, and make sure that I'm satisfied with the number of questions and all that sort of thing. It will be open, right? I'll open it up on Friday. It will be open until probably Tuesday at midnight, so that you have it done before Wednesday's class. What that means is you have to go in and do it in one block of time. You can't go in, log out, go in, log out kind of thing. But at least you have several days through. Hopefully at some point you can log in and do that. It will be mostly multiple choice, true and false kind of thing. I would say somewhere around 50 multiple choice, true and false. And then probably 2 short answer, like essay questions. But just by essays, I mean like 3 sentences, 5 sentences, not, not a paragraph, not 10, not 5 paragraphs or anything like that. I tend to do an essay question from each chapter, right? Just to kind of test your knowledge a little bit on that chapter. But I'll know the details. I'm thinking you'll have an hour and a half or whatever to do the exam. It'll be timed, and anybody that has accommodations, I'll put the accommodations into Moodle as well, so that you'll have extra time or anything like that you get for an accommodation. And I think that's about it. Are there questions that you guys have for me?
So the exam is on both the first 2 chapters, or it's just on chapter 2?
The first 2 chapters, chapters 1 and 2.
That
Chapter
lab
2
is,
has a lot of information in it.
Yeah,
I can't
exactly.
believe that's just
And-
one chapter. It's like,
Exactly. And
ooh.
that's why I do, when you look at it, there's like, I think 6 or 7 exams in this semester, but I'd rather give you an exam every couple of chapters, rather than trying to cram, 4 chapters into one exam where there's just so much information. Yeah. I think this is a better way to do it. Joshua, you were starting to say something.
yeah, I'm out of town right now. So
At the airport.
I was wondering when that lab is due. Friday,
No,
right?
the lab from last week is due Friday. Yep.
Okay, I'm gonna do everything when I get home tomorrow. Like I
Okay.
said, I've been trying to make it work, but that Mastering A&P website wants an Ethernet cable for some reason. So,
Oh, all right.
yeah, now I do it on my days off, which is Wednesday, Thursday.
Okay. All right.
But I'll knock all the stuff out when I get home tomorrow. Tomorrow.
Good.
All right. Yeah.
Sounds good.
I'm stuck here all day too. I could be doing it, but I don't want to do chemistry under pressure. That's why I got to be on that
Yeah.
one. I was like, I'm gonna do this real quick, and then I was like, no, I'm gonna stop. I'll wait. I'll redo it when I get home.
Exactly. And I should say this too. The first couple of chapters are harder than the rest of the semester, right? Nobody likes chemistry. It's abstract. It's not
I
anything you
do.
could, well, okay, I do too secretly, but it's very abstract, right? It's nothing you can touch. It's not like learning the blood vessels or learning the muscles or, something tactile like that. And so what I find often in A&P classes is the first couple chapters are the hardest, right? The students are trying to get used to me and get used to the class. And it's more abstract stuff. It's this chapter's chemistry, the next chapter, chapter 3 is cells, which again is really abstract. They're things that you can barely see with a microscope. So don't get discouraged by that, right? In my experience, grades always go up as the semester goes on, as you learn a bit more on how to study, and it becomes more of, you know, a system of the body. Oh, I can understand where the nerves are. I can understand where the muscles are. It becomes a little bit easier. So don't get discouraged if this is hard. This tends to be, I'd say, the chemistry and the cell biology chapters 2 and 3 tend to be the hardest chapters on students, which is another reason why I broke them up and put them on 2 different tests.
About the skeletal system.
Well, yeah, I mean, you know, you get to all those bones and the markings and all that kind of,
And the fossas, yeah.
they're all
that's
pretty
a lot.
lengthy. They're all lengthy, but I think you get used to how to study for it and the amount of material as well, you know.
All right.
All right. Well, if you have any questions tonight or tomorrow, definitely let me know and email me, and I'll try to get back to you as quick as I can. And I'll post this up there. Probably won't be posted up there till tonight. It takes, I don't know why, it takes a while for Zoom to like convert the recording to something that I can post. So I will post it tonight.
So there's no labs for this one, right? Or
There's
you're gonna-
no labs. I'm gonna try to find something for Friday that I can post that will be more of a just kind of a review that'll help you, you know, with the
Yeah.
test. But yeah, there's not really much of a lab you can do.
Not
For
gonna have us do
chemistry.
electrochemistry, pulling
No.
the rust off of a
Right,
copper
no.
onto.
And nothing in
Exactly.
toxins either, right?
What's that?
As far as I remember, I didn't see anything in that Pierce Parsons for work.
Right,
I
right.
don't remember seeing anything.
So
So-
there should be a quiz and a homework due, I think, next week.
Yeah. Yeah, there's a quiz next, the 21st, there's, I think, something due.
Okay. So I'm gonna go in there. I'll go in there today and just make sure that I'm, you know, make sure that I'm on top of everything. So just so you guys know, tomorrow, I have tomorrow off. And so tomorrow I'm gonna spend all day just making sure that this course and there's another one that I teach at GCC. I want to make sure that everything is set up for the whole semester, you know, that, I mean, of course, I'll probably have to make changes as we go, but pretty much set for the whole semester so that things are going forward are set. You know, so I'll be looking at those dates and trying to make sure everything's set. I can certainly email you tomorrow night, email everybody tomorrow night with, you know, going forward, this is what we're gonna do.
Okay.
Okay.
Alrighty.
All right.
Thing that helps make chemistry fun is if you draw this stuff out,
What's that?
practice drawing it. Making chemistry fun, I like drawing this stuff. Organic chemistry is like that too.
Organic,
Our
yeah, follow the
routines.
electron, right? I had a dry
right.
erase board and you follow the electron and you erase it and do it again. Oh, organic was the
Oh, yeah.
nightmare. I didn't like organic chem.
Really?
Did
Oh,
not like
wow.
organic chem.
Oh,
Could have been the
no.
teacher I had though, too. I don't know.
Yeah, I
That
think
makes a
that probably
difference.
makes a difference. But yeah. All right, cool.
All right. Well, have a good day, guys, and I will talk to you soon.
All right, take it easy,
Alrighty.
guys and girls.